Yes, so well, you are quietly sitting there for the next 30 minutes or so of my talk. You will have used about 10 liters of pure oxygen, and in the meantime, turned over about 1 kilogram of ATP. And this is all possible because of the respiratory chain in your mitochondria, who transfer electrons from NADH to oxygen, and in the process pump protons across the inner mitochondrial membrane. This proton gradient causes then rotation of the ATP synthase rotor and turns out all this ATP. And all these very complicated complexes of the respiratory chain have been studied in great detail in the last couple of years by X-ray crystallography, but also increasingly by cryoEM. We know a lot about them already, but the most questions are still about the most complex 1, which transfers electrons from NADH to ubiquinone and transports 4 protons across the membrane. This has been studied a lot by different groups in different species with increasingly good results. And in the last 2.5 years alone, there have been, I think, about 7 or 8 structures of complex 1 of different species at resolution better than 2.5 angstrom, where you can see thousands of water molecules. You can see all the individual atoms of the several iron silver clusters and everything else. So what we know about how it works is the electrons from NADH are extracted by the cofactor FMN, then transported over a series of iron silver clusters to the ubiquinone, which is located in a tunnel just above the membrane. And then somehow this triggers the transfer of protons across a series of conserved hydrophilic side chains in the middle of the membrane and lots of water molecules. And as I said, transports 4 protons. So we're learning a lot about how this actually works, but this is not the topic of my talk today. So what I will talk about today is how this very large complex of about 1 megadalton and depending on the species, more than 40 subunits is actually assembled. So some of the, many of the subunits are in the membrane and many in the mitochondrial matrix, some of them in the intermembrane space. And they all have to come together. And this is not a spontaneous process, but it requires at least 15 in humans assembly factors, which are proteins that are transiently associated with subcomplexes of complex 1. So a lot is known about how these complexes come together from complexome analysis, and which assembly factor is associated with which subcomplex. But the molecular details of how this works are almost completely unknown. And this we set out to study a couple of years ago. But it's difficult to do this in humans, of course. So we use a model organism, a yeast that can be genetically modified. And this is a long-standing collaboration in Frankfurt of the group of Volker Zickermann, who has been working with the Yarovia lipolytica complex 1 for more than 20 years. And with the Planck Institute, where I work, in the group of Werner Kühlbrand, where we do the cryo-EM. And much of the work has been driven by Christian, who has produced lots of beautiful structures, and he's now in Osnabrück. And later on, I will show a little bit of Christian's work. But for the first step, look at the PhD project of Jonathan Schiller, who looked at the first assembly intermediates and the AF1, which is known to be associated with the with one module of the protein pumping unit called the proximal protein pumping unit, shown here in green, and close to the phenome binding model. And here in colors, you can see the PP module in detail. So Jonathan knocked out the gene for AF1 in Yarovia and found that this strain only produces anymore the distal protein pumping unit, so showing that this really is an essential intermediate. And then he added AF1 back with a strep tag and affinity purified any assembly intermediates that will be associated with AF1. And then did cryo-EM of the intermediates. And this was in collaboration with Eike Lauber, who is the expert on cryo-EM of complex 1. So the first negative staining images showed very promising results, and class averages showed really distinct structures. So they went on to collect a large data set on the cryos, a cryo sample, about 6,000 movies. And Eike did a major processing effort, which I cannot talk about in detail for time reasons, but he found 2 distinct complexes which he could refine to a small intermediate at a resolution of about 3 angstrom and a larger one at a similar resolution. So how to identify this? So obviously this is the transmembrane part, and you could fit one of the core subunits of the membrane domain ND2, which is part of the PP module as expected, and one other small transmembrane unit. And the rest should be assembly factors. So an AlphaFold model of NDU of AF1 could be fitted easily. But then there was still a large structure left. So it is known that in humans, there are several other assembly factors associated with the ND2 subunit, shown here. But in Yarovia leucolytica, these are almost all missing, with one exception, the protein ACAT9. On the other hand, Yarovia has another assembly factor that is probably associated with ND2. So we had 2 candidates. CR84 we could fit into this structure. It is a very large protein, and it has a flexible arm that has no density, but we can clearly see that the structure is smearing out. So this is undoubtedly CR84. So then the other part should be ACAT9 on first sight. It looks possible, but we all tried to fit this and failed, and came to the conclusion that it had to be something else. And we had no clue what it was. But fortunately, the structure of the remaining protein was so good that we could, or Eike actually could identify a series of large side chains, shown here, and he did a search in the UniProt database of Yarovia proteins, and he found one hit. And that hit was a protein called tafatsin, which also came, of course, with an AlphaFold prediction, which we tried to fit. This worked, so we could model this protein and it turned out to be tafazin. So what is tafazin? It's actually a protein that you would not expect here. It's involved in the remodeling of cardiolipin, which is a signature lipid of mitochondria. And this protein had been identified more than 25 years ago as a protein involved in Barth syndrome, which is an inherited X-linked disease, which is characterized by muscle problems. So the Italian group who identified this protein named this after an Italian comedian called Tavazzi, who usually goes around hitting himself with a plastic bottle because of frustration. So probably the group had a lot of problems identifying this protein. But it was also pre-science because 25 years later, the structure of tafazin had still not been determined. So what does it do? It works on cardiolipin, which is an essential lipid in the inner mitochondrial membrane, and it's seen to be associated with all the proteins that we know, including complex 1, the ATP synthase, everything else. And the special thing is that it has 4 acyl chains. And the role of tafazin is now to replace during the synthesis of this lipid, the initially saturated chains by unsaturated ones, which is apparently essential for the proper function of the mitochondria. So what does it look like? This is the structure of the intermediate with tafazin here in green. And you can see that there's one long helix that is lying on the surface of the membrane. So this is apparently a membrane anchor that keeps the protein in a particular orientation. We could identify the active sites, which are to an histidine and an aspartate, which are conserved in acyl transferases, the family that tafazin belongs to. Also, we could identify 2 conserved arginines that are probably involved in the binding of the phosphates of the lipid substrate. And if you look now again at the structure of tafazin, you can see that the active site is oriented towards the membrane. So one possible function of the binding of this protein to the complex 1 assembly intermediate is to orient the active site in a proper way. Another possibility is not mutually exclusive, is that the binding of tafazin to a growing complex 1 ensures that cardiolipin is available where and when it is needed during protein synthesis. So what about the other assembly factors? The CR84 is the only protein that is bound to all the other proteins here, and it sort of has a central function. So the best guess that we have is that it sort of forms the core of the assembly intermediate, but yeah, other roles would have to be established later. And now what about AF1, which is essential in all organisms? That is not exactly clear from this structure, and for that we have to look at the large intermediate. So the large intermediate here has lost tafazin for the most part, but it has a lot of other density, and we can fit in here all the other subunits of the PP module. Shown here, but there's one gray density here that is left over that has not been identified. So is this another assembly factor? Well, when you look carefully, this is actually continuous with the orange subunit ND6. So doing a better fit to this, it is actually this helix that is left over. And also, if you look carefully, there's a little bit of extra density here inside the fold of AF1. So there's a clue here in this yellow helix, which is part of the subunit ND3. And ND3 has a very unusual topology. It consists of 3 transmembrane helices. The first 2 are located on one side of the membrane, and then there's a long loop. And then the third one here is on the other side. And actually, we were able to model this missing helix in the extra density. And this is now the model. And this shows that the specific fold of AF1 forms a binding pocket for the loop of ND3. So there's 2 possible functions here. One is clearly that it makes possible to assemble all the other subunits, especially ND1, because this would not be able to fit when ND3 was flying around here. Another possible function is that the ND3 loop has a conserved cysteine, and that could be protected in this way during assembly of the complex. And so here you can see the large conformational changes that have to occur during the further assembly of the whole complex 1. So what we know now so far about the assembly is that we first start with the ND2 and the C2 subunits with their assembly factors. Then the rest of the PP module attaches. And then during the further binding of the Q module, there are conformational changes of the, certainly the ND2 and the ND1 and ND3 subunits. And probably the Q module binds to the tether of the subunit A13, which is not visible in the assembly intermediate because it's flexible. Also, the PD module will bind, and this binds by means of the B5 subunit, which has a long tail going here under the PP module, where there's a perfect groove where this subunit will fit. So it comes in kind of like a staple and staples the 2 modules together. So what is now still missing is the N module here on top. And for that, we have to look at another bit of work that was inspired by a study a couple of years ago by Volker Ziekermann's group, where they looked at the S6 subunit, shown here in light purple. So it is known that mutations in this subunit cause lethal problems in. So complex 1 does not work anymore. And when they knocked out this subunit in Yarovia, they found that not only S6, but also the E12 subunit here in purple was missing. But instead, there was an assembly factor bound, AF2, which is related to A12. So this was the work of Christian Pareynau. He studied this the assembly intermediate based on the S6 knockout by cryoEM. So he processed a data set of about 1400 micrographs in Relion. It was quite straightforward. He got a beautiful structure that looked like a mature complex 1. But looking in more detail, you see that both the S6 and A12 subunit were missing, and instead there was another protein bound shown here in pink, where we could fit the AF2 assembly factor. So here you can see the structure of the AF2 and the A12, and the bottom part here consisting of a couple of beta strands and a short helix are very related to each other and bind to the same spot. But the A12 subunit has two ordered helices at the end terminus. It would be sitting here just above the membrane, whereas this part is completely disordered in the assembly factor. Then the middle part is quite different between the two, but the C terminus is again very closely related and forms the same structure. And this actually binds to the N module. So here looking at it in a bit more detail at the two structures. The already with the AF2 bound, the N module could come in. So this is probably the function of this protein. But why now do we need S6 to knock off this assembly factor and then replace it with a very closely related protein? So we think actually that the secret is in the two helices at the bottom, which in the A12 subunit form the tunnel for the substrate ubiquinone. And also there's some very highly ordered lipids bound to the mature complex 1. So probably the function of this AF2 is to prevent binding of ubiquinone while the N module is still not completely formed. So now to complete the story of the assembly, I think that the AF1 subunit will bind to this first assembly intermediate, then bind the N module to this C-terminal tail. Then S6 would come in and replace AF2 by the A12, and now we have the mature complex 1 that can be will be fully functional. So of course there's many more assembly factors, and we are planning on and are already actively working on looking at more structures of those and get more details about the assembly of the complex in Yarovia. And with that, I want to thank all the people involved in this. Most of them I have mentioned already, but I also especially want to thank our central EM facility led by Sonja Wells, who does an amazing job on keeping all our microscopes in tip-top shape, and Juan and Özkan, who make it possible for us to process the data. And finally, we are looking for a PhD student to continue on with this work. We have money from the DFG to continue this project. So if you're interested or know somebody who is interested, just contact me. And thank you for your attention.